Hello, my name is Corey McMillan, and I'm a research assistant professor in the Penn FTD Center. And today, with Dr. David Irwin, we will be talking researching in FTD and related conditions, why you should get involved in research. So we will cover in this talk what is research, approaches to research, how you can participate in research, and what you should consider when participating in research studies. So first, let's talk about what research is. We consider research that falls into two different categories. One is clinical research that can involve things like questionnaires, phone surveys, participating in neuropsychological or cognitive testing, or participating in imaging studies like MRI scans or PET scans. We can also think of research as biospecimens. This involves collecting biofluids like blood samples, cerebral spinal fluid through a lumbar puncture, saliva where we can collect DNA, and tissue, which can either come from autopsy or skin. It's important to consider what the difference is between research and medical care when deciding whether to participate in research studies. Research we can loosely define as any study that is performed in order to answer a question. So, for example, in clinical research, we do studies that involve people as participants, such as patients or healthy volunteers or family members. In contrast, basic research is research that involves the study of animals or cells that does not involve people. In contrast, medical care is specifically focused on the diagnosis and treatment of an individual. So in this chart, we review some of the key differences between research and medical care. One thing to consider is the goal. In medical care, the goal is to diagnose and treat an individual, where in clinical research, we're trying to answer specific health-related questions. The funding for these varies. So for medical care, the funding is supported through a patient or health plan, while in clinical research, funding is provided by a study sponsor, like the federal government or associations. In both medical care and clinical research, consent is required. So everyone participating, whether it's for medical care or clinical research, has to agree to participation. The assessments in medical care are based on patient needs, where in clinical research, they're based on a planned protocol. There are various protections that differ across different types of research. In medical care, state medical boards or professional standards are used to govern the protection of patients, where in clinical research, government agencies or institutional review board governs the protection of individuals. The certainty of procedures being performed varies. In medical care, they are medically proven, where in clinical research, they may be unproven and they may be exploratory in order to determine whether an outcome can affect medical care in the future. As far as access to information, in medical care, information is recorded in the medical records and in product labeling, while in clinical research, this may be exploratory data, and this may not be shared with individuals. And lastly, the release of findings is to the patient only in medical care, where in clinical research, we can typically release findings using de-identification in medical journals. So how do we go about performing research? So the traditional approach to therapeutic development we can view as a cycle. This cycle can start with data collection, and then from data collection, we then perform data analysis. From there, we can identify a potential target for medical care. Once we identify that target, we then have to perform basic science using animal or cellular models to sort of validate that what we're doing is successful. And then we need to identify biomarkers or tests to determine whether we're detecting the signal that we expect to observe using this approach. Once we have that information, we can design clinical trials to try to see whether the approach has an impact on individuals participating in research. And hopefully this helps the patient. Unfortunately, not all trials are successful, but we can learn a lot from this cycle. So what we learn from this can feed us back into different ways to collect data and different ways to analyze data and so forth, and the cycle repeats. Involvement of the patient in data collection is typically considered observational or natural history research. The analysis and target identification is typically considered dry lab research. The basic science and biomarker validation is considered wet lab research. And then the involvement in clinical trials we broadly refer to as clinical trial research. And importantly, you see that many of these boxes overlap because they're not all unique. They're highly collaborative and interactive with one another. So a typical example is that we can identify through DNA data collection a genetic target. So why are we interested in genetics? So from this example, you can see lots of different cookies. And these cookies all vary slightly in their sort of texture and the way that they look. And we can see, for example, that by adding a single ingredient, we can change the shape of a cookie. Or by substituting one ingredient for another, we can change the shape of the cookie. And then also by baking the cookies at different temperatures, we also get different shaped cookies. And we can consider this as a sort of environmental factor. In biology, the changing of a single ingredient can be produced by a gene. So we're interested in genes to manipulate ingredients and therefore make changes that can potentially affect an individual. The ways that we use genetic information varies. We can look at single alleles. So this is a single base pair of DNA. Or we can look at whole genes like C9orf72 that's involved in FTD and ALS. And over the past 2 decades, you can see here that the number of genes that we discover every year is rapidly increasing. This figure now is about 4 years old, but we're now at knowing about 40 genes that are involved in FTD. And the ultimate idea here is that by identifying a genetic target, we could then develop a treatment, and then that treatment would treat individuals that have that genetic variant. So for example, we could identify C9orf72 as an important gene, and then this would ultimately treat individuals that have C9orf72 expansion causing their FTD or ALS. In the FTD Center, we focus on precision medicine approaches to therapeutic development. So using the same cycle, we integrate multimodal sources of data that includes not just DNA, but also clinical research, biofluids and biospecimens, and also different types of imaging like PET imaging and MRI. And my lab focuses on data analysis of integrating all of these sources of data together so that we can try to identify novel targets for therapies. So for example, in C9orf72, one of the genes that causes FTD or ALS, we know that this leads to the development of C9orf72 pathology, and through processes that we're still trying to understand, like oxidative stress, this then leads to neurodegeneration. But importantly, what we've also identified is that there are epigenetic factors that can manipulate C9orf72. And what this does is this serves as a volume knob so that we could try to turn down the amount of expression of C9orf72 and ultimately reduce the aggregation of pathology and therefore hopefully reduce neurodegeneration. Ways that we've gone about validating this type of approach is using these multimodal precision medicine type approaches. So by looking at neuropathological tissue that patients have donated, we're able to see on the left sources of pathology in where the volume is turned down in C9orf72. And then on the right, we can see sources of pathology where the volume is turned up on C9orf72. And we see that there's much less brown spots on the right, suggesting that by turning up the volume, we're able to reduce the amount of pathology. We're able to find converging evidence for these findings using MRI. So in panel B, we see regions of the brain in red that are affected by FTD and specifically C9orf72. And then in the orange boxes, we see 3 brain regions that have appeared to be thicker in those individuals that are protected through this methylation mechanism. They include the hippocampus, the thalamus, and the motor cortex, which are all regions that are known to be affected by C9orf72. We can validate these findings in panels C and D by looking at the amount of neuronal loss associated with these in these brain regions. And then in panels E and F, we show that the percent of methylation affects the amount of longitudinal change. So in the colder colors, like blue, we see flatter lines, so slower rates of decline, where in red are hotter colors and faster rates of decline. So ultimately, by using this sort of multimodal approach, we hope that we're able to more precisely treat the heterogeneity of FTD and ALS. But next, I will turn to my colleague David Irwin, who will talk to you about how you can participate in research and what you should consider when participating in research studies.
Hello, I'm Dr. David Irwin. I'm a cognitive neurologist at the Penn FTD Center, and I'll be talking to you about participation in research from the patient's perspective and going over ways that patients and families can participate in research and some practical things to think about when considering participating in research, and giving you a good sample of questions that you may want to ask your doctor about the logistics of participating in research, which is a really important way for us to help advance the care and ultimate treatments for FTD. So how can I participate in research? There's many different types of research, and it's helpful to give a few definitions of the different forms of research and what's involved. So observational studies, these are studies that are often longitudinal, looking at the natural history of disease and collecting data and specimens from patients to help us understand the biology and help us frame how we would approach a clinical trial for FTD and related disorders. This is extremely important, as we know the biology of FTD is diverse, and for us to be able to understand what we can measure in the blood or spinal fluid, or what we can measure on imaging of the brain, and what we can measure with paper and pencil and sometimes computerized tests to test different forms of cognition, are very important, because ultimately these are the tools that help us make clinical trials for therapeutics most likely to succeed. So this is extremely valuable form of research. And at the Penn FTD Center, this approach has been taken for over a decade or more, where we've been collecting information with patients with FTD. And more recently, the NIH has prioritized large consortia, meaning other universities all working together, where we collect the same types of data and the same types of samples, so we can all work together as a team throughout the country and throughout the world. So this makes samples available to researchers who may have a great idea but may not have access to patient data or samples, and they can apply to the NIH. And this is a great collaborative way to grow research and to make advances more quickly. So the clinical types of research that are often involved in observational studies could include questionnaires about behavior or experiences at home for patients and families, a survey that could be done over the phone or through a video chat, neuropsychological testing. These are things that are often done similar to what's done in clinic, but looking at more exploratory tests to be more sensitive to some of the behavioral or language features of FTD. And then imaging studies, an MRI of the brain that is structural, to look at the structure of the brain and help us measure objectively areas of the brain that may be affected in FTD, so that we could use these as outcome measures in clinical trials. And emerging PET scans. These are scans that use a radio tracer that are specific to different forms of protein accumulations in the brain in FTD and in Alzheimer's disease. The types of biospecimens that are often collected, a blood sample, just like getting a regular blood test clinically. This blood sample can then be used to develop a blood test for FTD, would be an ultimate goal, where, as well as being able to do genetic testing from blood samples for research, looking at risk factors and other unknown factors that may help us explain why FTD is so different in different people. Cerebral spinal fluid is a relatively non-invasive biofluid. It's performed by a lumbar puncture that's done by a trained professional, a physician. And many patients equate this procedure to getting a dental procedure. This is done with a local anesthetic right in the clinic, and most patients feel vague pressure or a sensation of instrumentation without any significant pain. And this fluid, it circulates through the brain and the spinal cord and the nervous system several times a day, and it's usually very devoid of proteins or cells. So it's very sensitive for neurologic conditions where diseases of the brain may not show up in the blood, and it gives us very good complementary information to blood samples. And that spinal fluid, we know we can measure proteins related to Alzheimer's disease like tau and A-beta, and there is some data that these proteins may have meaning for diagnosis and prognosis in FTD as well. And this is a rapidly growing area of research and really vital to a lot of the work that we do. Saliva is another biofluid or biospecimen. This is a non-invasive way to get DNA for genetic testing, for example, and then tissue and brain donation. Brain donation is a critical part of research for FTD. As you know, FTD is very heterogeneous, meaning there's different pathologies that are actually under the microscope that's caused the very same or similar clinical syndromes. And your doctor can diagnose FTD or primary progressive aphasia, but it's only until, without looking at the brain under the microscope, we don't know if it's the specific form of FTD caused by the tau protein or the TDP43 protein, or sometimes Alzheimer's disease plaques and tangles can cause an identical clinical syndrome as FTD. And having brain tissue for donation is a critical part to validate or help us understand the data that we collect during life. And it's an extremely meaningful contribution to research. And at the Penn FTD Center, we provide a formal neuropathological report from the neuropathologist of the final diagnosis and review with families after that report is generated on the meaning of those results and so forth. So these are an overview of the different types of clinical research that patients can participate in. Clinical trials or therapeutic trials are a different type of research. This type of research tests an intervention or a therapeutic that may help improve the condition. And there's been a sea change of new clinical trials for FTD, and we're guardedly optimistic about the rapid growth in this area. And there has been efforts to help accelerate the development of therapies for FTD. And this overview shows the normal trajectory of how a specific drug or intervention is developed for clinical trials. And there are preclinical early studies that are done in animals and cell models to look for the biologic rationale for a therapeutic. There is human safety data in what's called a phase 1 study. So these are studies looking at healthy people for safety of the agent. And then there are expanded safety called a phase 2, where we're involving more people, and that could include healthy people or patients with disease. And then there's what is a phase 3, which are large multi-center trials whose goal it is to ultimately look for a clinical benefit. And that's what would go for the FDA to get approvals. And there are legislations to help accelerate this process because of the urgency for need for therapeutics in FTD. At the Penn FTD Center, we are involved in several clinical trials, including observational studies where we look at imaging and the MRI and PET scans in patients with FTD and with ALS, as well as other neurodegenerative diseases like Alzheimer's disease and Lewy body disease. We have an app development for apathy, a very troublesome feature of FTD, the lack of motivation, and this app program helps caregivers and patients create a schedule and prompt for more interactive activities during the day. We also collaborate with Dr. Ben Kim in the ophthalmology department, looking at the back of the eye with a non-invasive scanning technique called OCT. This involves a visit similar to going to the eye doctor, where there's a dilated eye exam, and this non-invasive way of scanning the retina is a direct connection to the brain. And we think that there are differences that we may be able to measure in a non-invasive way that could tell us about the biology of FTD and Alzheimer's disease, and potentially tell us about diagnosis and prognosis. Clinical trials have all different types. There are symptomatic trials. These are trials aiming at the symptoms of FTD, and we have collaboration with Dr. Roy Hamilton in the neurology department, something called TDCS. This is a non-invasive way to stimulate parts of the brain that are involved in language for patients with primary progressive aphasia. Patients with stroke who have damage in the language parts of the brain, this non-invasive technique of adding a very small amount of current to the scalp can stimulate healthy parts of the brain to compensate and help with word finding and fluency or the rate of speech. And we've been testing this in primary progressive aphasia, and this is available to patients. And we're also involved in several disease-modifying therapy trials. So these are different. These are attacking the biology of the disease with the ultimate goal of trying to slow or stop the disease. And currently we have a clinical trial for progranulin families to targeting the mechanism of progranulin genetics with the development of FTD. We've recently completed a trial looking at tau, an intervention to stop the tau protein in progressive supranuclear palsy patients, and we'll soon have similar trials for PSP and for patients who have familial FTD involving the tau gene. And we'll soon also be having a trial for Alzheimer's disease patients targeting amyloid. And there are several other trials planned targeting the mechanism of disease. So to move on to some practical issues of what to consider when participating in research, it's important to be informed as a patient and a family member. So who can participate in research at the Penn FTD Center? Everyone. Symptomatic patients. We study patients with the behavioral variant of FTD and primary progressive aphasia, as well as the progressive supranuclear palsy and cortical basal syndrome. We also do observational research with patients with different forms of Alzheimer's disease, dementia with Lewy bodies, and ALS. So this research is critical for us to help understand the biology of the disease, know what types of tests that would be the best to detect a change in a clinical trial. And these studies are ongoing and open. We also welcome healthy volunteers, and this can come from caregivers or family members of patients with FTD or related disorders, and also community members, people in the community, neighbors, friends who would like to spend the time with us. Having healthy volunteer data is also very critical for us to compare what we measure in patients to healthy adults so that we can help make these tests better for clinical trials. As a participant in clinical research, it's important to be informed about what's involved. So good questions to consider talking to your doctor with include, what's the purpose of the study? Why is the study being conducted? What tests or treatments or procedures will be involved? Is this an observational study? And if so, does this include an MRI, a lumbar puncture, and other procedures? If this is a therapeutic trial, there are often similar collections of that type of data. So understanding what the timeline of activities and what's involved are helpful for you to make the decision to participate. What are the risks and what are the potential benefits? This is something that we weigh very carefully, and everything that we do in clinical research is carefully balanced to make sure that any intervention or procedure that we do for research is balanced with the potential benefit for the patient. So observational research is very low risk. The procedures that we do have low risk, but very large benefit for the FTD community for us to help know more about the disease. And therapeutic trials go through rigorous review at the FDA to ensure that the risks of a potential agent or study drug or procedure are balanced by the potential benefit for the patient. And these are important things to go over with your doctor. And then how long will the study last and how frequent the visits are. Our observational research at the Penn FTD Center is focused around visits to see our physicians clinically. So we try to minimize the extra transportation back and forth to the center and build it into the schedule of the day where you'd be here anyway to see us clinically. For therapeutic trials where there is a treatment being given, this often requires extra visits to the doctor for safety, to check for extra visits that would be outside of your clinical care. So these are things to consider for logistics of different aspects of research. The design of clinical trials are important to be informed on and to know a few definitions. So most therapeutic trials are placebo-controlled and blinded. What that means is that most therapeutic trials have a group of patients who are receiving a placebo or not the study drug. And this is often blinded where your doctor, and the people in the study, all the staff are blinded. We don't know who's getting the placebo and who is getting the study drug. And the reason for this is so that we can ensure that there's an objective measurement. We can see a potential benefit. And most studies in FTD and related disorders are designed to really maximize the number of patients that receive the active drug and use the minimum amount of people who have to get their placebo to see an effect. And as an extra benefit to patients, most trials have an open label period. This means that once the part of the trial that is testing for a benefit, where some patients are getting placebo and other patients are getting therapies, the open label segment of the trial then allows for everyone to get the study drug. It's important to know that there's a screening visit that determines eligibility. You may want to participate in a clinical trial and it may not be feasible. And this could be for safety reasons. Most clinical trials have specific inclusion and exclusion criteria, meaning clinical features or blood test parameters or other health issues that you'd have to meet those criteria to be able to safely participate. So that's the reason for those designs. And it's important to follow closely with your physician. Trials are designed to maximize safety. But with study drugs, these are in a phase that's experimental, and we want to make sure that we are monitoring things closely. So when in doubt, it's always a good idea to reach out to your doctor with any new symptom. What are the risks and benefits of participation? So all clinical observational research, this is the research that collects data and samples from patients. There's typically no direct benefit, although coming to the center and interacting with our team often gives opportunities for socialization for patients and interacting with our social work team and others that many patients enjoy. And there's typically minimal risk. These procedures, including the lumbar puncture, are considered low risk, and this is carefully monitored by our institutional review board. So it's extremely meaningful for research and for the goal of developing therapies for FTD with very minimal risk. Clinical trials, there may be a benefit for disease-modifying or therapeutic interventions, including targeting symptoms. Interventions have variable risk depending on the treatment, but in general, these are minimized and are carefully selected through a process through the FDA and our local institutional review board. So it's very important to discuss these aspects with your doctor to make the decision that works best for you. We'll end with acknowledgments. We have a large team at the Penn FTD Center and integrate with our colleagues at the ALS Center and at the Center for Neurodegenerative Disease Research, as well as other centers for genetics and imaging at Penn and consortia for ALS at the University of Miami. And we have federal funding from the NIH to do these types of studies. And most importantly, we are very grateful to patients and families who volunteer their time to participate in research. And I'll end with giving you some useful resources and websites, including the website for the Penn FTD Center and our colleagues at the Penn Memory Center and Center for Neurodegenerative Disease Research, and the Association for Frontotemporal Dementias, which we've worked together with to make a informative brochure about genetics of FTD, and affiliated advocacy groups for all different forms of Alzheimer's disease and related disorders, as well as clinicaltrials.gov is a wonderful resource that lists all the clinical trials in the US, and you can search by diagnosis to find clinical trials that you may be eligible for. Thank you very much.